One of the reasons we're doing these experiments is that osteoarthritis and degenerative diseases of cartilage is really a severe health problem in this country. Like 40% of individuals over the age of 40 have some sort of cartilage degeneration, which results in immobility and pain. And we want to use human embryonic stem cells that have differentiated into chondrocytes to try to elicit repair.
So it's possible, we don't know, but it's possible that directing human embryonic stem cells into joint cells might be another approach to derive appropriate chondrocytes for joint repair.
Chondrocytes are the cells which comprise the cartilage of the joint. In order to promote the differentiation of human embryonic stem cells into cartilage, we try to mimic the events which regulate cartilage differentiation in the embryo. The first step in that process in the embryo is that cells, undifferentiated cells, sort of similar to embryonic stem cells, become closely packed with one another and communicate with one another. So what we did was take the human embryonic stem cells and put them in a culture environment in which the cells are closely packed together in a small amount of medium. And in addition to these cells cultured at high densities, we've added some of the signaling molecules that we've learned regulate the differentiation of cells of the embryo in the chondrocytes. And under these high density culture conditions, along with the signaling molecules, we get uniform differentiation, it appears, of the human ES cells into chondrocytes. To our knowledge, we've developed some protocols that have not been reported as yet in the literature, where we can directly obtain chondrocytes from the human ES cells. That has not yet been reported. From examination of the literature, we're getting more uniform differentiation than others have reported. Most of the studies are done with adult stem cells, stem cells from the bone marrow or stem cells from other parts of the body. The human embryonic stem cells have a lot of advantages over those cells for repair, we think. These are not published, okay? They're not peer-reviewed, but the data is solid, and I'm very confident in the data. We don't know whether it's the chondrocytes at different phases of the lineage, early in the lineage, in the middle of the lineage, late in the lineage, or if we were able to get joint precursors specifically. Yeah. Whether, we don't know what will work, but
it'll be fun to find out.
It'll be fun to find out, and I think we have a good opportunity.